Today I would like to tell you a story about a new healing mechanism that we found in our lab. And my name is Ron, by the way. So imagine that you are bored, you're at home, it's raining outside, very bored, nothing on Netflix, so you decide to play soccer at home. Bad idea. And you hit the window. Okay, so what are you going to do? So first of all, I assume that you will cover the hole in the window because you don't want to get the rain inside. And similarly, in our body, when we injure ourselves, like a scratch or whatever, the first step is forming a blood clot that helps you to distinguish between, to keep the inside environment from the outside. Then the idea is to clean up the mess, the broken glass. And similarly, in our body, we have the hero cells from the immune system that comes over to eliminate the damage by an inflammatory process. Okay, so now we need a new window, right? So we're going to call the window shop and ask for a new window. And in our body, cells are invading into the injury site and proliferate. They are dividing in order to replace the lost tissue. And eventually we install this new window, and in our body is the remodeling phase, which we are trying to get back to what it was. And in general, from the definition of healing is the ability or the process of the body to maintain the structure and function of the tissue. Together, these steps are part of the classical healing steps. And in general, in our lab, the Zeltzer lab, we are very interested in the musculoskeletal system, and more particularly a tissue called the tendon-to-bone attachment, or the enthesis. It's located all over our body, where we need to connect between tendon and bones. And in general, if you need to think about how do you connect between these distinct tissues, we have tendons on the one hand that are elastic and bones from the other. What you're going to do? You're going to super glue them? It's not enough. So nature had a very sophisticated solution, which is to form a gradient between these two elements. And while it is important, it is also prone to injuries. And unfortunately, it never fully heals. The current solution for this kind of injuries is to suturing the tendon back to the bone by using threads and nails. And this is not the elegant transition between them, and it tends to be to fall apart again. So in general, we also know that some have better healing capacities than others, for instance, babies or young versus old or mature, as I prefer. And so we thought that maybe we can utilize these inherent abilities to find the healing, better healing capacities of the enthesis. So the idea was simple. We're going to use mice, young mice that are 7 days old, and perform an injury on the Achilles tendon. And I will elaborate a bit more about this structure. So we have the tendon on the left, cartilage on the right. We performed an injury, and in general, this tissue have a limited number of cells, not much cells, a lot of extracellular matrix, which is proteins. And this tissue has also limited access to blood and oxygen. So with that being said, we made an injury to the enthesis, and we were expect to see the classical attributes of the healing process. So, but what happened is that when we performed the injury, there was no bleeding because there is no blood access, so no bleeding. Then we waited for several days. We really looked in multiple time points, and there was no inflammation. It means that no cells entered the injury site. But so what happened instead? what we found is that the cells themselves, the enthesis cells, the resident cells, they produce collagens. Collagen is the most abundant protein in the body. And tendon and cells usually produce collagen type 1, and cartilage cells, chondrocytes, often produce collagen type 2. And together, each zone, we're able to create its a signature protein. And that leads us to a formation of a scar that is a new type of scar because it doesn't have any cells inside. And it was able to fill the gap like a roba on your kitchen. And we could observe that there were no proliferative cells. Opposite, we saw that the cells that are in the adjacent tissue are gone. In the tendon, they are absolutely gone. And in the cartilage end, you can see ghosts of cells. What used to be cells, they remain there until eternity, like Eshet Lot. I'm sorry, I don't know the name. And eventually, this scar was present even 1 year later. So there was no remodeling. And also unbelievable that I waited an entire year to tell you that. So the injured enthesis forms a permanent acellular scar. But you can say, okay, Ron, you said that that healing is the ability to restore structure and function. So, okay, we restore the structure, what about the function? So we conducted the behavioral tests when we measured the paws of the injured animals, and we saw that 2 weeks later they had an impaired gait capacity, which was over time able to regain it. So it means that the enthesis were able to restore its functional properties. So just to summarize things up, this is a new healing mechanism. And why is that? Because first of all, we don't see any attributes from the classical healing model. So this is a non-inflammatory process. It is mediated by the local cells themselves, opposed to cells that comes from, other places. The heroes are the cells, and these cells are sacrificing themselves to produce this large amount of collagen proteins to form a scar, to form a plug. And eventually the enthesis is able to restore its function. So I would like to thank you all for listening, but mainly to Neta Felsenthal that initiated this project, my collaborators, my fantastic lab members, my advisor Ellie, and for you for not, bailing after lunch. So thank you so much.
Thank you very much, Ron, for this interesting talk. Questions, please? Yes.
Did you see if there were perhaps any effects still there? For example, were these mice perhaps more susceptible to being injured again in the same location? Or because you showed that it restored the function, but was there any other long-lasting effect to this kind of scar?
This is very interesting. Actually, I don't know. We did histology for many time points afterwards, and I didn't see any major effect, but there's always a possibility.
Was healing different in young mice and old mice compared to young mice?
Yes, when I tried to perform this injury on adult mice that were 1 month old, we didn't see any formation of the cellular scar. It was just a hole that remained there. So I guess this is a neonatal thing.
Have you characterized the properties of the scar tissue? Is it elastic as the collagen or as the tendon, or no look at that?
I don't know. I try to characterize it by using staining for types of molecules, but I got collagen type 1 and type 2, couldn't find anything else. So, but when we did electron microscopy, we saw that the order of the fibers were very misaligned. So I don't know what is the properties, but I know it's not the same as it's supposed to be.
Did you check that in vitro as well?
No, no.
More questions? Yeah. Do you see any damage to the nerve?
I haven't looked, but this is interesting. No.
Because you saw that the mice are not walking properly, so you think it's only the leg? Or
So in this, I didn't show you because I didn't want to load too much, but we did also a sham injury, which we had, we mimicked the injury but without the needle punch, and there was no effect. So I assume the injury had an impact, but I don't know about the nerve level.
So let's thank again, Ron. Thank you very much.